we will be starting our session today with the voice from the community, and we'll be again introducing our video from one of our community members, Daniel Webb.
G'day, my name's Daniel and I'm the lead JDRF advocate down here in Tassie. I was diagnosed with type 1 when I was 6 years old back in 1995, and about 7 or 8 years ago I was also diagnosed with diabetic retinopathy. At the time this involved some pretty invasive treatment, and it's not a matter of if but when I require further surgery. But within the last 4 or 5 years, with the advancements in CGM and insulin pump therapy, my time in range has improved dramatically to 80 or 90% per day. My HbA1c has dropped really well and I generally feel a lot better. And this means that further complications with my retinopathy and other diabetes related complications are being prevented. And for that, I want to thank everybody there today. I know that there's been a lot of research being done into treatment and a cure. And for the first time in a long time, it gives people like myself optimism and hope that our long-term outlook with type 1 will be a positive one. This is a big change in my mindset for the first 25 years of having type 1, and for that I want to thank you all. So it makes a huge difference in our lives. So thank you.
I would like to now introduce our chair of the next session, Dr. Avani Hines, who is a senior research fellow based at the Telethon Kids Institute in Perth. She leads the epidemiology research in the Children's Diabetes Center and is also investigator at the JDRF Global Center for Excellence. Welcome, Avani, and please.
Thanks, Dorota, and welcome everyone to, I'm sure, what's going to be a very interesting session this morning. So it's my privilege to reintroduce Professor Mark Atkinson, who, as you know, is sadly unable to actually be with us in person today. Mark, as many of you know and might remember from tomorrow, is from yesterday, is an American Diabetes Association eminent scholar for diabetes research and the Jeffrey Keane Family Professor at the University of Florida. He's currently the Executive Director of the JDRF Network for Pancreatic Organ Donors with Diabetes, or nPOD, program, and he recently completed his steering committee chair of the NIH Human Islet Research Network. So we're now going to listen to a kindly recorded presentation from Mark titled Lessons from nPOD and the Human Islet Network.
Hi, I'm Mark Atkinson. I've been asked to provide a brief lecture regarding lessons learned from nPOD, the Network for Pancreatic Organ Donors from Diabetes, and the HPAP portion of the Human Islet Research Network. So in preparation for this presentation, or this former presentation, I went on the newest thing, the ChatGPT, and asked it a question. I said, how will studies of the pancreas tell us how type 1 diabetes develops? And independently, you can see that ChatGPT said studies of the pancreas are essential in helping us to understand how type 1 diabetes develops. Keyword being essential. And if you read on, it tells you that it'll have therapeutic implications, biomarker implications, and more. So big thumbs up as an early user of ChatGPT. Now, this should not be too much of a surprise because, again, much of what we understand about diabetes has been derived from long-term or many years of studies of the human pancreas, dating back all the way to Langerhans with the islets and understanding of variant forms of pancreas and different forms of diabetes. But what happened was in about 15, 16 years ago, after not seeing as much translation as we'd like to see, we developed this program called nPOD, which is to obtain tissues from organ donors with or at various risks for diabetes and study them for what they might tell us about the disease using more modern techniques. Soon after, the NIH developed a new program called HPAP, which is a parallel effort, except that HPAP is involved with more data generation and making it public, whereas nPOD is more of a distribution network. Both of them have grown large, And we're very proud, and hopefully at the end of this lecture, you'll see why we're happy with the results that have came from them. So one of the first ways that these programs have been of help is to improve our understanding of how the disease develops and overturn some dogmas. So for example, people often say, even to this day, that type 1 diabetes occurs when there's 85 to 90% of beta cells destroyed. But actually, in looking at new onset cases of the disease with modern techniques and having the whole pancreas, we know that the beta cell loss or the degree of it is quite variable amongst individuals. Another thing is that it's been told many times over that within a few months to years of type 1 diabetes onset, you lose all of your beta cells. That also is not true, in that we can look at pancreas from people who've had type 1 diabetes for decades, then they still have a small amount of residual beta cells, so it never goes to completion. Another thing that we're seeing is great diversity in terms of pancreatic histology, which leads us to believe that perhaps it's not a singular disease, but a cluster of diseases that have commonality of a need for exogenous insulin replacement. And then a major impact, and again, it fits well within this section, on the notion of beta cell replication. When does it occur and how does it occur? And I'll touch on those points in a few moments. Another long-standing dogma is that insulitis is a chronic and predominant feature in the natural history. Actually, insulitis is very hard to visualize in the pre-diabetic stage, even if you only have, the entire pancreas. And we're trying to understand the reasons why that. At onset, 100% of people have it, but the percentage of islets that in the pancreas that have it and when it occurs is quite variable. We also have suffered in a way from a lack of intensity in terms of research on beta cells in the past. And I'm going to show some data later in the talk that will increase the potential role for these cells in the disease, and the whole notion that it all around the disease only revolves around islets and beta cells. I think we're going to show data that there's the story goes beyond that. So let's start with this first puzzle of piece of the puzzle, if you would, of what is insulitis and what can we learn about it beyond what was learned 50 years ago. And in order to do this, I'm going to introduce a whole series of new techniques that unfortunately I don't have time to explain on the right. But the main point I want to make here is that in the past, we were reliant heavily on more traditional means of histology, H&E staining, immunohistochemistry, some immunofluorescence with limited number of markers. But now there's a whole bevy, and the list grows yearly, of new technologies that will give us expanded insights on the disease. So one of the first things I want to share is that the notion that the lesion itself is a mixed bag. You can find almost any cell of the immune system present within the insulitis lesion. And as I noted, for some reason, despite the insulitis that's so predominant at onset, in rare cases do you actually destroy all of the insulin producing beta cells, even as noted, up to 50 years after disease onset. And why this is important in terms of the composition of the insulitis, there's been some recent work in the last 4 or 5 years to suggest that the presence of insulitis in its composition varies by the age of onset. And this work shown from a group of UK investigators demonstrates that if you develop type 1 diabetes at a very early age, say age 7 or 8 or less, you have a predominance of B lymphocytes or CD20 high cells, whereas as you move, say, individuals who develop type 1 diabetes later in life, the B cells are essentially absent. So this suggests that, again, there may be different pathogenic mechanisms in play as a function of age of onset. And we've used some modern techniques just within the last year to show this. This is using a technique called Codex, where we use multiple antibodies. And if you look across the top, there are 2 cases of individuals at onset who developed the disease at 3 and 4 years of age. And you'll see the red cells in the top, which are indicative of the B lymphocytes. But if you look at the sections in below, of those that develop disease in the 20s, you don't see those B lymphocytes. However, in all cases, you see the CD8 positive cells. Another thing that we were encouraged to see is that we've been doing things right in terms of the autoantigens, meaning does what we measure in peripheral blood reflect what might be going on in the pancreas? And we believe, at least in terms of autoimmunity, the answer is yes. Here's two different ways of analyzing the question. One is to do staining with tetramers and look at the reactivities of cells within the pancreas. And you see that, again, the markers of autoantibodies seem to be present in the pancreas. And the second way on the right-hand side is from an investigator at Harvard, Sally Kent, and her group, where they've cloned T cells out. This has been done by Stuart Mannering and others in Australia, and tried to take a look at what the reactivities are in terms of autoantigen specificity, and you see the list there, and they do represent what we've been studying in peripheral blood. So we seem to be on the right path here. So let's use some new techniques here and look at insulitis in three-dimensionality. And what you can see is that insulitis can vary from islet to islet, and in here, the magenta color is staining for insulin, and then the gold color is for CD8 cells. And you can see sometimes there's a patching, there's a clustering, and sometimes there's an envelopment of the islet. So there's a variation across the islets in terms of what you see. Now here's some newer data using a three-color immunofluorescence that also takes into account the alpha cells and their involvement in the disease. What you can see here, again, magenta being the insulin-positive cells, CD8 cells in yellow, and then the alpha cells in blue. And what you saw there was, again, great diversity in terms of the way that the immune cells and their population seems to target the cells. Let's take this another level forward, where we call this a fly-through. On the bottom, you'll see insulin cells stained in pink, and the CD8 cells in yellow. And on the top is what we call a flip chart. And you can progress from the top of an islet down. And this is in a normal individual. So this is what we see as a normal composition of islets moving through the pancreas. However, if we switch up and go to a case of type 1 diabetes, uses the same technique. Now, the standard immunohistochemistry on the left-hand side, that's what you would see. But now when you do fly-throughs or the flip charts, you get a much better feel of what the lesion looks like in multi-dimensional analyses. And we hope over time to use this to actually identify the question of why some islets are destroyed, how they're destroyed, and more. We've also been able to use HPAP and nPOD tissues to understand about islet destruction in vitro models, where we take pancreas from individuals with the disease, cut it into small slices. And on the panel in the middle of your slide there, you see comparison to a control case on the left-hand side, where, again, CD3 cells are stained in yellow, and then the insulin beta cells in the magenta color there. And you can see there's a lot of activity that goes in and around the islets. And let me make this clear. This is pancreas sections obtained from somebody, and the analysis was done with no manipulation approximately 24 hours after the pancreas was isolated. And we think this is going to be a great way for drug discovery and understanding, again, mechanisms of disease. Another thing that we've observed in type 1 diabetes is a lobularity, that if you go into the pancreas and look, you'll see that there's lobes that are where there's no islets that contain insulin-positive cells, as is shown in the left-hand side here above the red line. And then there's islets on the right-hand side or lower left portion where there's a lot of yellow, and that's the CD8 infiltrate. So if you compare west, no, versus east, yes, side of that slide, you'll see the big difference of demarcation. And now we really are ramping up the notion that insulitis predominantly affects insulin-positive cells. They do not like insulin. Another feature of the pancreas in type 1 diabetes that has been described in the past is this hyperexpression of class 1 MHC, suggesting that the islets or the beta cells themselves are stressed, and especially, again, if they're insulin containing. And that's what's shown on the right-hand side quantitatively, that compared to controls, type 1 diabetes islets that are insulin containing have high levels of MHC expression, whereas in those islets that they're not, it's a little less. So all of this is in support of an autoimmune nature of the disease, and I'm not here to say that it's not at all. But the central tenet is that autoimmunity occurs because you have a loss of central and peripheral tolerance mechanisms, and you see autoantigens. However, there has been in the background a long time this theory put forward initially by Gianfranco Battazzo, excuse me, back nearly 45 years ago, where he was questioning whether type 1 diabetes resulted from a, quote, homicide of the islet or a suicide of the islets, meaning homicide, immune cells going in and just destroying the tissues, versus a suicide where the beta cells themselves contribute to disease. And there's been a few write-ups since that have expanded on this question. So let me turn now to what we've learned from studies of the pancreas with respect to islets and beta cells. And there's a lot of data emerging on this. So one of the first things that we and others have been questioning is why do you see this mosaic? Why are there different types of islets within the pancreas of somebody with type 1 diabetes? Islets where they're completely devoid of insulin, islets that look, at least on their surface by histology, to be normal, and why are other islets inflamed? And all three can exist in a pancreas at the same time. And so one of the ways we have done this, both in nPOD and HERN HPAP, had been to use tissue cytop. And I'm going to combine some studies here where this is published and unpublished data. But the bottom line is we've taken pancreas sections from nearly 100 cases where they've been divided up into non-diabetics, people in the natural history of disease, recent onset or long-term cases. And again, I apologize, I don't have time to explain cytop, but in principle, what it is the ability for us to detect utilize nearly 90 different antibodies at the same time on a particular tissue slice. And these are the types of antibodies in the classes what we look at them by. And from this, we've been able to make a number of observations. And one of them is if you use some high-end data analysis tools, you can see that actually what we observe to the eye as being normal islets in recent onset cases, they're not exactly normal. They carry many functional variations compared to normal islets. And on the right-hand side, you can see, using these tools, a description of what a control islet would look like. On the left, in the middle, in the orange, is what a new onset type 1 diabetes case looks like. And on the right, an islet from somebody with established disease. And another thing we've been able to use tissue cytop for is to identify when in the natural history do you begin to lose the beta cells themselves. And what you can see here is actually it's a relatively late process in the natural history of the disease if you compare single antibodies to multiple antibodies to recent onset cases. And this recent onset case by tissue cytop also validates what I said earlier about the great variability of beta cell mass at the time of disease onset. Another variation on analysis with using tissue cytop is the ability to look at changes over time in the natural history. This is a very complex slide, and I apologize for that, but let me just draw you to the left-hand side and say that as you move from the control situation to new onset, there's a series of molecules that appear to decline in their expression. Well, there's a few others that increase. And then there's some that we continue as we do our analysis to look at that are suspicious, but we can't quite make calls yet. Another thing that tissue cytop has allowed us to do is to be very exact in terms of where class 1 upregulation occurs. And what we've been impressed with is that actually it's not restricted to the beta cells, but if you look across the different groups, you see hyperexpression in alpha cells, delta cells, ductal cells, and even in the acinar cells. So that we need to understand more about that upregulated class 1 MHC. And again, our field is moving so much towards single cell analysis and 3-dimension analysis. One of the things we've been very excited to do is to join with investigators at Johns Hopkins and create this new methodology that's called CODA. And it involves artificial intelligence and training to understand histology. It's a very brute force and laborious technique, But through it, with time, we hope to again be able to share with the community single cell, three-dimensionality, starting with pancreatic blocks, but eventually build out to the whole human pancreas and be able to tell at amazing resolution what changes are occurring in this organ in the natural history of diabetes. Now, again, fitting within this section, one of the other things we've been intrigued about and why beta cells may be involved in the disease is their number. And unfortunately, there's a conception that almost everybody has the same amount of beta cells if you don't have diabetes in life. We now know through studies of nPOD and other biorepositories that that's not true, that there's variation amongst normals. The other thing that we've been able to do with nPOD is shown in the right-hand side, where we have been able to show that replication rates in normals over time, and that it appears like a situation where by the age of 2 to 4, you are essentially locked in the number of beta cells that you have. And it's almost like the growth curve. And with that, another individual, Dr. Battaglia, and I developed this concept about 7, 8 years ago, and we still think it may hold true, is that in early life, the amount of beta cells that you have will set you up at various levels of risk for type 1 diabetes. And this is a factor that may be unappreciated. And then one more thing I'd like to highlight about beta cell aberrations and why they may be important. And unfortunately, I don't have time to list all of these, but there are now some 24 publications in the literature that, taking a look at nPOD or HPAP tissues, have identified a series of aberrations that are present in both the beta cells, as shown on the panel A here on the right, comparing to normal situations, and then even in the islets. And these are in a publication that will soon be out in Cell Metabolism, and I'd encourage you all to look at it if this is of interest to you. And why this is important is we're beginning to grow in our belief that the model we have for staging of type 1 diabetes needs to be modified, and that it's not just stage 1, 2, and 3, that there may be a stage 0.5 or stage 0, if you would, that predates the multiple antibody phase that is marked by those beta cell and islet cell aberrations I showed in the earlier slide. And then the last 2 minutes, I'd just like to focus on another part of the puzzle that we think that these two programs have brought in, and that's the role of the exocrine pancreas. In most textbooks and literature, people think the exocrine pancreas is not involved, but we began to notice as we collected these tissues, situations like in the left, we have 2 individuals that are matched for age and BMI, but the pancreas of an individual with type 1 was half the weight. We've now gone on to study this in hundreds of individuals with their pancreas weights by collection the organ donors. And again, you can see that the type 1 diabetes pancreas is in fact smaller. And people sometimes criticize studies of tissues and say, what about living subjects? So we went on to do a study analyzing over 250 individuals at various stages of diabetes, and found that in blinded analysis with board-certified radiologists, it is true. The pancreas of an individual with recent onset type 1 diabetes is quite small relative to a age and BMI match control, but even those at risk for disease and relatives without autoantibodies appear to be smaller. And this is also reflected in serum or plasma if you look for pancreatic enzymes. So we, again, back to that cell metabolism paper I'd encourage you to look at, we feel like there's also a natural history of the pancreas that occurs with changes in whether or not these exocrine changes impact the islet or the immune system is a hot question right now. And this is a paper that's just out on bioRxiv by the Bluestone and Boakley group that, again, comes to the conclusion that it's the exocrine pancreas that provides a framework for what insulitis will look like. So we have a lot of future directions here. We're going to move beyond pretty movies. We're going to do things called co-registration where we combine techniques. We're going to increase the use of AI, and we're going to push more for seeing HPAP and nPOD studies move toward biomarker and clinical development. I hope you found this presentation interesting, and I thank you again for the opportunity to present.
So hopefully you all enjoyed that. I think, Mark's talk was excellent, and lots more things to think about and some new paradigm shifts. So next, it's my privilege to introduce Professor Natasha Rogers, who's giving a keynote talk titled Tailoring the Success of Islet Transplantation. Natasha is a senior staff specialist at Westmead Hospital, where she's head of transplantation and director of the clinical islet transplant program. She's also Professor of Nephrology and Transplantation Medicine at the University of Sydney, where she's an NHMRC Emerging Leader Fellow and Deputy Director of the Centre for Transplant and Renal Research at the Westmead Institute for Medical Research. Thanks, Natasha.
Thank you so much. Good morning, everyone. Thanks very much for the opportunity to speak today. So before I begin, I'd just like to acknowledge the traditional custodians of the land in Western Sydney, where much of the data that I'm going to show you has been generated, and like to pay my respects to the elders past, present and emerging. So I'm going to really just go through our islet transplant program and what we offer in Australia and our results, as well as some translational research. So many of you will be familiar with the Diabetes Control and Complications Trial, which was published now 30 years ago, where if you improve your HbA1c, you minimise your risk of microvascular complications, including retinopathy as well as nephropathy, but pair that with an increase in severe hypoglycemia rates. So hypoglycemia, severe hypoglycemia, is not insignificant in the type 1 diabetic population, and a small proportion of people will require intervention by medical personnel. And so there's indeed a small subset of type 1 diabetics who have repeated episodes of severe hypoglycemia. And of course, if you're hypo unaware, your rate of events of severe hypoglycemia increases almost exponentially. Hypoglycemia comes with a mortality risk, and whilst it pales in comparison to the risks associated with chronic kidney disease or cardiovascular disease, there is a defined risk of death. So in Australia, we offer islet transplantation to treat type 1 diabetes, and this can restore near normal glycemia. It most importantly protects from severe hypoglycemia, and in a subset of patients will also provide insulin independence. So there's 3 centres in Australia that will perform islet transplantation, and we can isolate islets at 2 centres in Melbourne and in Sydney and transport them to Adelaide for transplantation. And so this is just a composite histogram reflecting the activity in the 3 centres around Australia. But of course, it's a national program, and we accept national referrals from every state. So the program was developed at Westmead Hospital under Phil O'Connell and Wayne Hawthorne's expertise, and was a journey over 20 plus years, beginning with preclinical studies leading to large animal models, a successful human islet isolation preparation, and the first clinical islet transplantation in 2002. Of course, many of you would be familiar with the islet isolation procedure, which is long, laborious and complex. And down in the bottom right-hand corner, you can see islet operations managers dressed up like that for about 8 hours to complete a preparation. And we know that islet isolation is hard work. And this is a composite graph from the ANZIPTA registry, and it shows that a large proportion of islet isolations are not proceeding to clinical transplantation, and so a ratio of about 3 to 1 of islet isolations that are discarded versus transplantation. We did manage to have a bumper year right before the COVID pandemic. So we employ the use of the interventional radiologist to access percutaneously the portal vein and infuse the islets into the patients, as you can see there. So this is one of the preliminary publications from the National Islet Transplant Consortium, published now a decade ago, and this is a composite value of the first 17 patients, and what you will see, the patients are listed along the x-axis and with their Edmonton hypo scores along the y-axis, and then along the z-axis coming forward in time. So the blue lines at the top, at the back of the graph, are going to be their hypo scores pre-transplantation. You really see that flattening out over time post-transplantation. And so when we looked at this in terms of subsequent follow-up as part of the ANZIPTA registry, what you can see is their improvement in hypo score following the first transplantation. And that really flattens out after the third transplantation, so you can have a maximum of 3 islet transplantations performed separately. But I'll just draw your attention to the fact these are small patient numbers. And there's significant variability in the C-peptide, despite excellent response in terms of hypo score, and less fluctuations in HbA1c. In keeping with international standards, we do achieve insulin independence in about 40% of people, and this is long term, but again, really small numbers of patients. So this is some work performed by our endocrinologist at Westmead Hospital, Jane Holmes-Walker, who compared meter-dose injection with a continuous subcutaneous insulin infusion and islet cell transplantation at 6 and 12 months. Keeping in note the mortality associated with severe hypoglycemia, there were 5 deaths on the wait list, and when you look at hypo score, standard deviation in glucose, and continuous overall net glycemic action, there's substantial improvement following islet cell transplantation over and above what is achievable with some of the newer technologies related to insulin infusion. So our current immunosuppression protocol associated with islet transplantation, we have induction with thymoglobulin, really annihilates your T cells, and standard immunosuppression, for those familiar with these agents, is tacrolimus and mycophenolate. Now, tacrolimus is a problem. It's a calcineurin inhibitor, and calcineurin inhibitors are associated with a nephropathy. So this is an old study, but still holds true and demonstrates that over time, regardless of what your primary transplanted organ is, and that's the same for cell therapy, in addition to solid organs, over time, you increase your risk of renal impairment. And some people do indeed proceed to require renal transplantation as a result. So we moved to look at a new immunosuppression protocol, same induction regimen, but the use of belatacept, which is a CTLA-4 immunoglobulin fusion protein, and sirolimus, which is an mTOR inhibitor. So for those who are not immunologists, this is the antigen-presenting cell T-cell interface required for allogeneic immune cell activation. And so you require process through signal 1, signal 2, and signal 3. So our standard immunosuppression protocol is calcineurin inhibitors, which inhibit NFAT, which is a transcription factor, and mycophenolic acid, which impacts on nucleotide synthesis and limits cell cycle proliferation. However, our new immunosuppression protocol intersects at signal 2 through CTLA-4, and the mTOR inhibitor also impacts upon cell cycle signalling. So belatacept's important. It's unfortunately not freely available in Australia, so we have BMS to thank for this drug. So, as I mentioned before, it binds with very high affinity to CD8 and CD86, which are maturation markers present on antigen-presenting cells, and delivers an inhibitory signal such that rather than activation, you actually get T-cell energy, and also promotes development of regulatory T-cells. So we performed a study where we had head-to-head comparison of patients receiving tacrolimus and mycophenolate, as well as belatacept and sirolimus. And so these are just some preliminary results over the first 24 months. Again, small numbers of patients, but really importantly, there's good safety and feasibility, and you can see comparatively similar changes in terms of daily insulin requirement, C-peptide excursion post-transplantation, HbA1c, and significant reductions in their hypoglycemia score, which is encouraging. Now, when it comes to graft outcome measurement, we looked at this in terms of the Eagles criteria. And so in those receiving tacrolimus and mycophenolate, you can see there, we've got a proportion of people with good or even optimal long-term outcomes. We had fabulous results early on with our belatacept sirolimus immunosuppression protocol at 3 to 6 months. Unfortunately, we commenced this study prior to COVID, and that had a significant impact in terms of our ability to retransplant patients and improve long-term outcomes. So we're also interested in whole blood immunophenotyping. I'd just like to acknowledge some of the senior scientists in the lab, Elvira and Min, who are in the audience, and they developed this protocol where we can look at a number of leukocyte subsets from a really small amount of blood and follow these longitudinally. This is quite complex and took a significant period of time to develop. And so we have 7 leukocyte profiling panels, and you can see the complexity there. and it takes a whole team of people all day to sort through this staining, and you get panels like this, which we can then interpret. So I'm just going to show you some preliminary data. So once again, we've compared our Baladicept sirolimus patients with tacrolimus mycophenolate mofetil, and you can see here that if you're on Baladicept and sirolimus, you have increased FOXP3 Tregs, increased naive T cells, and changes in your central memory and effector T cell population. So we think this is potentially significant in terms of graft outcome. So many of you will be very familiar with benefits of islet transplantation, and we have a number of challenges. One is a shortage of donors, and I'm very interested in how islets don't function optimally after transplantation. So I'm going to just take you through some translational research that my lab is conducting. So I'm very interested in the cell surface receptor CD47. It's widely expressed. In fact, it's expressed on every single cell, so it's got a broader range of expression over even class 1 HLA because it's present on red blood cells. So it has a number of binding partners, including SERP-alpha, and many of you might know about CD47 in terms of the don't eat me signal. It also binds integrins, and it has a soluble ligand thrombus 1-1. So rather than having an immunological focus related to CD47, we know that it's a cell signalling protein and affects cell adhesion, motility, apoptosis. My lab's been at the forefront of looking at aspects of CD47 signalling in acute kidney injury, blood flow and angiogenesis, but its role in metabolic disease is unexplored. So we're very interested in whether CD47 was expressed in the endocrine pancreas, and this is just some immunofluorescence from wild type and CD47 knockout animals, and you can see that whilst CD47 in the green is expressed everywhere, it has increased expression in the endocrine pancreas, and you can see the histogram on the right-hand side. So when we isolate islets from wild type or CD47 knockout animals, you can see there's increased insulin expression via this Western blot. When we perform an intraperitoneal glucose tolerance test, these are global knockout mice, and I understand that that response is multifactorial, but you can see improved glycemic profile in the knockout animals. When you put them on a high-fat diet, they don't seem to gain weight, so we can move over Ozempic. Similarly, when you treat wild-type mice with a single dose of a CD47 blocking agent, you can really recapitulate the changes in glycemic response to a glucose load. So we're very encouraged by that. So then we wanted to have a look at ultrastructurally whether these islets were significantly different. And what you can see in the CD47 knockout islets, you have an increased number, particularly under high glucose concentration, increased number of islets that are docked at the plasma membrane. You can see that they're within 100 nanometers there. And we can also visualise exocytosing insulin. This is not in real time, so these are static pictures, obviously, but under high glucose concentration in the CD47 knockout islets, you can see increased exocytosis. So we're also interested whether CD47 plays a role in diabetes, and so we looked at female nod mice actually and isolated islets, and you can see CD47 expression increasing with time as this mice developed overt diabetes. And we could actually intersect this process by giving weekly CD47 antibody administration. You can see the improvement in glycemic profile there. And when we looked at their islets, the MYAP301, that's the clone of CD47 antibody, can see actually islet hypertrophy, but without any significant change in terms of insulitis. So we think we're actually modifying beta cell function. Finally, in terms of our islet transplantation studies, this is a syngeneic model, so there's no immunosuppression. When we transplant CD47 bearing islets into STZ-treated black 6 mice, we see some minor decrease in their blood glucose levels, but when we transplant the same number of 47 knockout islets, we see marked euglycemia. And when we treat wild-type islets with a single dose of CD47 blocking antibody and then wash the islets and transplant them, we get significant improvements in blood sugar level. So I think I'm going to leave it at that. So I'd just like to acknowledge the National Islet Transplant Consortium and centres in Sydney, Melbourne and Adelaide I'd also like to acknowledge my lab, in particular Arthava Kale, who performed much of the experimental work. If everyone's looking for a great postdoc when she's finished a PhD, she's sitting over there. And also acknowledge the funding that supported some of this work. Thanks very much.
Thank you, Natasha. So next up, again, it's my privilege to introduce you to Professor Shane Grey. Shane is currently head of the School of Biotechnology and Biomolecular Science at the University of New South Wales. He's also head of the Transplant Immunology Research Team, director of the Functional Genomics of Type 1 Diabetes program at the Garvan Institute of Medical Research. He was lead investigator on Australia's first clinical islet transplant program and program director of a national NHMRC JDRF research program for type 1 diabetes and a founder of the Australian Islet Study Group. So we're very, very lucky to have him here. Thank you, Shane, whose presentation is titled From the Silk Road to Human Trial, Gene Therapy for Type 1 Diabetes.
That's the last time I'll ask my mum to write my intro. So... But anyway, thank you very much. So yeah, I'm going to have a very loose allusion to the Silk Road and in this story, and it is a bit of a journey. And I thought, a few years ago, I was lucky enough to travel some of those paths, so I thought I'd just intersperse the talk with some photos from my own experience. So let's go. So as we just heard from Natasha talking about the progress of the Australian islet transplant program, I think we can all see there are benefits for beta cell replacement, including islet cell transplantation. The current practice, as Natasha described, is intraportal into the liver, but there are a lot of challenges with it, right? We all recognise that, these good outcomes for patients with hypoglycemic unawareness, but, these limited supply of islets, which potentially could be overcome by stem cells, and stem cell trials are occurring, and also the need for heavy immunosuppression. So these are the real roadblocks to help us roll this out to more patients who would benefit. And what the end result of this talk is to get to a position where we're going to test this idea that we could tune down immunity at the level of the islet graft by engineering them with a gene called TNF-ARP3. So I'll show you. So let's start the journey and we'll see how we got there. Yeah. So it starts one part of the journey, and these are a couple of parallel roads which all interweave. One part of the journey started with a clinical program that I'm a part of at the Garvan Institute, and includes Sydney area health hospitals, called the Clinical Immunogenomics Research Consortium of Australasia. And the aim of that consortium is to use whole genome sequencing and to identify causal genes in children with very complex immune disease, to try to identify a candidate gene which then can be drugged. If we cannot associate a drug to the causal gene, then it goes into a research setting. And in this case, 3 probands from 3 families presented with an unusual patterns of immune disease. Just to give an example here, we say that child 1 from family 1 here presented with early onset fevers, mouth ulcers, inflammatory bowel disease, eye inflammation, and you can see an image of the eye inflammation from child 1 there. Graves' disease and autoimmune serology. Patient 2 had many of those same features, but also type 1 diabetes. So you can see really complex manifestations. What was interesting about these patients was all 3 of them showed a deletion in the TNF-ARP3 locus. So this is a map of how we see the TNF-ARP3 locus from a genetic perspective. The black boxes are the exons, which code for the protein. And you can see that patient 1 had what we call deletion 1. The red line is the whole locus is missing. Patient 2 had a premature stop codon mutation here in exon 6. And the third patient had a 3' UTR deletion, which potentially would upset translation and lead to loss of protein. That was interesting because a few years ago, or just a couple of years before that, this paper had come out showing that loss of TNF-ARP3 through deletions led to a disease which was called A20 haploinsufficiency, which looked a little bit like Behçet's disease, which is an unknown autoimmune condition which includes eye inflammation, mouth and genital ulcers, and has also more recently been associated with kind of things that we would think of as more common autoimmune disease, so sort of overlapping the phenotype of the 3 children. And I guess this is the one allusion to the Silk Road. Another big breakthrough, this disease is not really understood. If you pick up your medical textbooks, there's one paragraph on it. And it's quite a bad disease in the sense that patients can quite often go blind. There's no real treatment for it. But one interesting discovery was made in 2013 was that it's found most predominantly along what we would call the old Silk Road route. And part of the explanation for that is it associates with a particular MHC haplotype that is prevalent along this, trade route. And so the discovery that TNF-ALP3 deletions would also contribute to disease was a big breakthrough. But when we went, the other thing that was kind of important that we knew about TNF-ALP3 is that it's absolutely essential to control inflammation in vertebrates. So here a group knocked out TNF-ALP3 in mice. We also mutated it to make it non-functional in mice and also deleted it in zebrafish. And you can see all the animals that are deleted, they're all runted, they all show body malformations, and all these mice suffer from premature lethality. So they all die by about 2-3 weeks of age after birth, and they all suffer from uncontrolled sepsis. So you absolutely need the A20 through vertebrate evolution, or the TNF-ALP3 gene, to control inflammation. And it does that by acting as a ubiquitin editing domain. That's the only biochemistry you're going to get, a protein. But it does cut ubiquitin chains, if you want to know, with this domain here. And it also adds ubiquitin chains with this particular domain here. And that's important because things like TNF and IL-1 and TLRs signal through cascades where ubiquitin plays a big role. This kinase, RIP1, is required for TNF to signal activation of what we call the NF-kappa B and AP1 transcription factors. Don't glaze over quite yet. It's not going to get much more difficult than that. But those transcription factors are important for immunology because they literally control hundreds of immune genes that are regulated in immune effector function, recruitment, graft rejection, autoimmune disease. One of the other things that the NF-kappa B system controls, though, is TNF-ALP3, producing the protein which we call A20, and this OTU domain cuts off those ubiquitin chains and then adds another ubiquitin chain that causes the protein to be degraded. So that's how it sort of shuts down the system. If you miss this protein, you end up with this kind of hyperacute immunity to endogenous pathogens. So coming back to our patients, we thought, okay, QED, they've got haploinsufficiency of A20, but as we were sort of looking at the data, if you can read, you can see here that the DEL in each case, DEL 1, 2, 3, was also found in a parent, and the parents were otherwise healthy with no history of autoimmune or inflammatory disease in the families. And that was a real mystery. So now we've got an immunological conundrum. What's going on here? So we went back to the genomic data, and what we noticed was that every single one of the probands also carried in cis this pair of coding variants in exon 2 and 3 of A20, T108A and I207L. And we also noticed that these variants were also carried in healthy parents, so there's a deletion, the haplotype we're calling it, and there was even a healthy parent who was homozygous. So what is this variant doing? So what we did was did some biochemistry. So we cloned, so we've got the I207L T108A haplotype in this O2 domain, one of these important enzymic domains of the A20 protein. This domain is interesting because there was a family reported with Behçet syndrome, which also carried a missense variant in this domain. And there's a GWAS hit for autoimmune disease that's also coding in TNF-ALP3, and that's also in this domain as well. So we sort of cloned all these things. We actually also found a mouse in a forward genetic screen that also had a hyperimmune phenotype with a missense variant in this domain. So we cloned all these variants and then tested in what we call an NF-kappa B reporter assay. We're just literally saying TNF-ALP3 turns off NF-kappa B activity. How well did the variants do that? And when you rank them all, we saw this really interesting pattern. So in blue, as you see that we call the wild type A20 function, the anti-inflammatory function, we normalize that to 100%. And when you look at the children who carried the T108A I207L variant, you can see that their A20 is still suppressive, but about 15% less. Was that really significant? We'll get to that in a second. But we also saw that there are other variants, such as the variant associated with Behçet syndrome in a Japanese family was much more strongly loss of function. So there is this sort of evidence now for the first time that rather than being a binary on-off switch for immunity, there are these graded control points appearing in the TNF-AIP3 gene that might change its function. So that led us to go back to our patients, and we said, well, that would predict then that they might have a higher stimulus response. So we took PBMCs from patient 1 and treated them with TNF and just asked, do we see what's the expression levels of NF-kappa B dependent genes? Here we're looking at ICAM1 and CXL2. You can see the green shaded boxes here. They represent the sort of normal range of expression we get from healthy PBMC donors, but the red line indicates the child with both the deletion and this kind of unusual pair of A20 missense variants. You can see they're hyper-responsive to a TNF stimulation. So they're starting to look like they're carrying a hyperinflammatory phenotype. So that led us to this sort of hypothesis then. So A20 variants do show different capacity to control inflammation, and inflammation obviously governs the tempo of an immune response. So if your A20 function was increased, we'd expect your immune system to be cooler, right? You'd have relatively more anti-inflammatory effect. And if your A20 function was decreasing, you get an increased inflammation. So to test this idea, we generated a whole pile of mice using CRISPR editing technology, where we actually introduced then these different A20 variants into the germline, and then started to ask the question, how would that affect their immune response to a pathogen trigger? So for instance, just to remind you, and this mouse was particularly carrying the I287L variant from the children. And so one of the things that we did was challenge the mice with an enteroviral infection. And this was interesting thinking of the context yesterday and the potential association of enteroviruses with T1D. But in this Kaplan-Meier survival chart, the purpley data here, in the blue, that represents wild-type mice, littermates, receiving a challenge of the enteroviral infection, which is basically 100% lethal. But if you're carrying the I287L variant as a homozygous haplotype, you can see that these mice are actually showing some protection against a lethal viral infection. And if you step it up by using another mouse, remember this is the mouse that had the slightly stronger loss of function variant that was in red, you can see these mice are 100% protected from the virus pathogen, and there's even a heterozygote advantage. So this is showing in real time that at a biochemical level, there are variants that reduce A20's function. They give you a stronger stimulus response. We saw that in patient PBMCs. And when we introduce that into mice, we can see in real-world immunology, this leads to a boost of your immune system. So this sort of really made us start thinking, could these children be carrying a version of A20 that boosts their immunity, and that's contributing to their hypersensitivity, their reaction, their autoimmunity, and their sort of disease phenotype. And so to test that in a real sort of much more immunological way, we actually made mice now that were representing the family. So we have the I287L variant mice. We made another mouse that had a deletion, so haploinsufficient. We crossed the mice, and what we found, which was really remarkable, I don't know, you don't really need to know much pathology, but if you look over here on the far left, that's a normal colon from a healthy mouse. This is now the mouse that has both the I287L and the deletion, and you can see that the colon is completely disrupted, and these mice are presenting the type of pathology and phenotype that we're seeing in the patient. So they've got a gut phenotype, for instance. So we could actually have some causality in terms of understanding the disease. There's a lot of immunology there, you don't care. But what's really interesting about this is we discovered that A20 deletions are not always fully penetrant, and I guess that's part of this problem of, Mendelian diseases. Are they really Mendelian? What are all the cofactors? In the case of these children, the cofactor turns out to be carrying a allele of TNF-AIP3, which has a slight reduction in function, which presumably boosts their threshold up of immune stimulation over some sort of checkpoint, which leads to disease. And I think the really good story is that, of course, now working with the clinicians, there are drugs that target this pathway. So we can actually say to them, anti-TNFs and so forth could be beneficial for these patients, which is what's happening. The other thing that was really interesting, from a population perspective, we actually found that this variant only exists in Oceania. So the red and purple here indicates the frequency. Basically, the blue color shows that there's no representation of this allele anywhere west of the Wallace line. So this suggested to us, combined with the fact that you've got a boost in your immunity and a protective response against some pathogens, this suggests that this is an evolutionary conserved variant that's been, selected in Oceania to provide an immune boost for protective benefit. So the important thing for this talk, because somehow we're going to get back to a transplant, is that this is telling us that nature is using TNF-alpha-3 to change how strong your immune response is. That's the main point here. That's the aha moment. Stay with me. So if nature's using versions of A20 to make your immune system more or less aggressive, why can't we? That could be kind of interesting therapeutically. So, back on the road. That's Annapurna 2, just touching 8,000 metres, going up the Marzi Gandhi Valley, somewhere we're going to cross over there in a couple of weeks. So in a parallel thing, we've been interested in why is it that the clinicians use the same techniques essentially to match organs and patients and retrieve organs, the same kit of drugs, but different organs survive differently in a transplant setting. Why is that? And we know that islets particularly have a poor survival rate. You know, because we generally think that the immune system, and I'm not changing dogma, does attack the graft. I mean, that's fairly true. But if we're if we're drugging the immune system, why do we still get these variable outcomes? Is there something else to the puzzle that we haven't quite picked up on? And we've sort of been thinking about, is it possible that there's more of a dance? That there is something going on with the graft? It's not just a full one-way street. Have we missed something? And then missing something means there's a new opportunity that we could think about. Is the graft a therapeutic opportunity that we're not realising? So part of the Australian clinical program that Natasha just nicely presented previously, one of the things we did was we asked the question, what do the islet cells look like from an immunological perspective as they're going into the patients? And here's an islet prep, and then we used at the day microarray technology, which was the thing to do. One of the kind of really impressive and possibly shocking things was that islets are literally loaded with pro-inflammatory factors. So all the red here, all these things, these are chemokines that recruit every possible immune cell that you definitely do not want at your graft site. But the interesting thing is we probed along into these sort of pathways and mechanisms and looked at the early response of islets to this kind of trauma. We actually saw they were making other things. They were making TNF-alpha-3. It's one of the most highly expressed genes in an inflamed islet. And we just heard that TNF-alpha-3 is absolutely important for controlling inflammation. So we thought, what is that doing in the context of a graft? And so one of the first things we did was we took advantage of the mouse we had that had the loss of function TNF-alpha-3 variant. And we said, well, okay, so that immune system of that animals we've talked about would be more aggressive, more prone to an inflammatory trigger. What's going to happen to that graft in a transplant? So the model is an islet cell transplant under the kidney capsule into a diabetic recipient, and it's a full MHC mismatch, so it's a very strong donor reaction. The blue line represents, let's say, one sort of wild type A20, if you will, going into a normal recipient mouse, and there's the rejection tempo, so you can see all mice reject these allografts. But if you would carry this particular version of A20 that's a loss of function, so it has reduced anti-inflammatory function, the islets are actually more rapidly rejected. And you can see this when you biopsy the grafts. In the red again is the islet cells that are having the more hyper-inflammatory version of TNF-alpha-3. They are indeed expressing lots more inflammatory factors, more IL-6, more interferon gamma, more TNF, more CXCL10. So this gene is having a role in the graft. So what about if we went the reverse? So in this experiment, what we did was we used an adenovirus to force the expression of high levels of A20 in the islet graft. The very, very unfairly small chart on the top right basically is showing you it's a number of chemokines, CCL2, CXL10, CXL1. It's showing that when you force the expression of A20 in these islet cells, the third column is where A20 is expressed, and you can see that it's suppressing their ability to be inflamed. When we transplanted islets with this into diabetic recipient mice across, again, a full MHC mismatch barrier, all the control grafts were rejected really rapidly, and about half the A20 expressing grafts survived forever. These mice died of old age and normal glycemia. The islets at post 100 days were beautiful. This is the histopathology of the grafts, beautiful H&E and lovely insulin staining, just looking like an islet in a pancreas. Interestingly, there are these little inclusions of immune cells. And when we investigated that further, we found that these turned out to be FOXP3 regulatory cells. And through a whole lot of immunological studies, we could actually show just by forcing the expression of A20 in the graft, we were able to induce a graft-specific tolerance that was permanent. So, coming back to the start, we've got this problem. We've got this interesting idea that beta cell replacement is an interesting opportunity for preventing long-term complications and providing good metabolic control on a daily basis. And we've got, what is this? Is this a Sopwith camel? Is this the kitty hawk? Where's Colin? I loved your analogy about the Air Force. So where are we, right? So how can we take it forward? Could we use what we've learned here to genetically engineer islet cells with A20? So this is the real mountain that we're going to climb, right? Let's go over it. So we've just been funded for an MRFF mechanism through what they call the Targeted Translational Research Accelerator to actually test the idea of restoring glucose control in T1D patients with genetically engineered GARV AAV2 A20 islet cells. And this will be a first-in-human safety and efficacy trial. This is the team that you absolutely need. No person is an island, and when you want to do these things, obviously we need the whole Australian community. I definitely want to thank Phil and Tom for really providing platforms for us to do this. And Toby will lead the clinical arm of the trial over at the Royal Adelaide Hospital. Ian Alexander is important as our gene therapy expert, and Daniel Barton will help us commercialise it. And so am I completely over time? The plan this is work of Toby Coates, where he's established in his research facility over in the RA, this fantastic and very innovative way to transplant islet cells into the skin in an intradermal site that's prepared with a matrix. And in the green, you get this beautiful vascularization of human cells in the pig skin, which is a model for human skin. And here you can see a patient lining up to have the incision prepared for their islet cell transplant, and a patient has received this procedure, and Toby will for sure talk about that. So our plan would be to add into this skin dermal transplant site the gene-engineered A20. So basically the idea is A20 will suppress this inflammation at the graft site and promote this sort of Treg expansion and tolerance. There's a long way to go, right? So now we're in Australia. This is an Australian road. I've travelled down in the Nullarbor. We've finished the vector design. We've started process development. I didn't even know process development was a word 2 years ago, but it's a thing and it consumes my life right now. Human ethics has begun, patient recruitment has begun, and Toby's begun his control studies, as we call them. And there's a long way to go, but hopefully we'll get there. This is the team who did everything. Thank you. Sorry for being over time.